Hi everyone and welcome back to ByHollyG. Welcome to today's video. So this one, as you can tell from the title, is all about antibiotic resistance, and the reason why I'm choosing to talk about this is because first of all, I feel like we've all been prescribed antibiotics at some point in our lives, and secondly, I truly believe that antibiotic resistance is a huge problem, but not many people know that. You know, it doesn't gain enough attention, and I do recommend watching my video on bacteria before this one just so you get a bit of background knowledge, because obviously antibiotic resistance is all to do with and very much linked to bacteria. So as usual, I hope you enjoy this video. Definitely give it a thumbs up if you do. Comment down below, subscribe if you are new for more biology, and we will dive straight into the video. So the first question is obviously, what are antibiotics? And they are essentially substances that can be used against bacterial infections, and that really should be emphasized. Like, they cannot be prescribed for like viral infections. They are only effective against bacteria. And the first antibiotics, so the penicillins, they were discovered in 1928 by Alexander Fleming. They are derived from fungi, but we now have loads of other antibiotics. So we have like streptomycin, vancomycin, amoxicillin. You know, there are so many antibiotics. And something else we should say here is that we divide antibiotics into broad spectrum and narrow spectrum. So broad spectrum antibiotics, they will target a wide range of bacterial species, whereas narrow spectrum antibiotics will only target a few of them. And another way we can classify antibiotics is based on their kind of mechanism. So are they bacteriostatic or bactericidal? And the bacteriostatic ones, they don't kill bacterial cells, they just halt their growth and proliferation. So they kind of like prevent the growth of a bacterial cell population. And then in contrast, the bactericidal antibiotics, they will actually kill the bacterial cells. And that nicely leads me on to the next question, is how do antibiotics actually work? And the answer to this isn't really so straightforward. They can act via a variety of mechanisms, but what's really important is that they must achieve, just like any drug, selective toxicity. And that means that an antibiotic, it must only target the bacterial cells and not, say, for example, the human cells if the antibiotics are being used in our bodies. And that is normally quite easy to do with antibiotics because bacterial cells, and if you've watched my previous video, they are prokaryotes, whereas human cells are eukaryotic. So there are lots of differences between those two cell types, and we can target therefore the bacterial cell only and leave the human cells alone. So antibiotics, they usually target, say for example, the cell wall, so the peptidoglycan cell wall of bacterial cells, components in their cell membrane, the 70S ribosomes, which are in contrast to 80S ribosomes in eukaryotes, the bacterial cell genome, bacterial proteins or enzymes. You know, there are a range of structures, so we can achieve this selective toxicity. A specific example then of a particular mechanism that many antibiotics use is through preventing the cross-linking of the peptidoglycan in the bacterial cell wall. That is just a very common mechanism, and peptidoglycan, as I talked about in my other video, is this complex polymer, and yeah, the antibiotics basically just interfere with the synthesis of the bacterial cell wall. So with that background knowledge, we're then going to start by looking at antibiotic resistance specifically. So basically, what is antibiotic resistance first of all? In short, this is basically where bacterial cells are no longer affected by the antibiotic. And this is essentially a process of evolution by natural selection. So in any population, we will always have genetic diversity. So there will always be genetic differences between the bacterial cells in a particular population. And normally that diversity is just caused by random mutations, and those are just changes to the DNA sequence. But regardless, we always have this genetic diversity. And sometimes a particular mutation might mean that a bacterial cell is resistant to an antibiotic, so it is no longer affected by this drug. And so if we apply a bactericidal antibiotic to that population, most of those bacterial cells will die. However, we might have that single cell, or that group of cells maybe, that all have the same mutation. They're left over, they're not killed. And because we now have a very small population, and because the competition for like space and nutrients is very low, it means that those few bacterial cells that are remaining can just grow and proliferate and expand quite rapidly. And as that population grows, that gene that leads to the antibiotic resistance will spread first of all by vertical gene transfer. So that just means through the generations. So as that bacterial cell divides by binary fission, it will pass on its genes, and the new daughter cells that are made will inherit that resistance gene. However, alongside that vertical gene transfer, there will be horizontal gene transfer, which we're going to talk about next. But this is a very powerful means in which mutations and genetic information can spread between bacterial cells, and horizontal gene transfer basically describes the spread of genetic information that's not in a parent to offspring manner. So for example, like me just passing my genes onto you through the screen, that might be like horizontal gene transfer, so not like down the generations. But my main point here is that this resistance gene will just spread very quickly, so you eventually end up with a whole population that is resistant to the antibiotic, and that spread can take place vertically, so vertical gene transfer, and by horizontal gene transfer. So that's, for example, looking at resistance to one antibiotic, but then you can imagine that situation happening over and over again as you apply different antibiotics. So we've already talked about horizontal gene transfer, and I just wanted to expand a bit on this because, as I said, it's very powerful in creating genetic diversity within bacterial cell populations, and it can therefore drive the emergence of antibiotic resistance, just because it means that genetic information can spread very rapidly and very easily between different bacterial cells. Not only can genetic information be passed between like very closely related species, but also very distantly related species by horizontal gene transfer, just because of this huge fluidity and the ease by which this occurs. New bacterial variants can arise really quite easily, and this emergence of new variants is not constrained to, you know, random mutations, natural selection, and vertical gene transfer. So as I said, horizontal gene transfer, it is this spread of genetic information between cells or organisms that are not in a parent to offspring relationship, and it's not just constrained to bacterial cells, but here we're obviously talking about horizontal gene transfer in the context of bacterial cell populations. And this intercellular movement, so the movement of genetic information between bacterial cells, that takes place in three main mechanisms. So the first two, which we're not going to talk about in detail, are transformation and transduction. So transformation is just simply the uptake of naked DNA from a bacterial cell's surroundings. It has been observed that bacterial cells can, you know, induce the killing of a certain bacterial cell so that they release their DNA, and then that bacterial cell that induced the killing can take up the DNA by transformation. That's not really frequently seen, but we have observed it. And then transduction is basically the transfer of genetic information that's mediated by what's called a bacteriophage, and that is simply a virus that infects bacterial cells. And the third mechanism of horizontal gene transfer is then conjugation, and this is the one I'm going to talk about in the most detail, just because it's the most efficient and the most powerful mechanism of horizontal gene transfer, as we'll see. So conjugation, we'll define it basically as this unidirectional transfer of genetic information between two bacterial cells, so in an intercellular manner, that is mediated by a transient, so a temporary cell-to-cell interaction. And what's really important in conjugation is the pili, or the conjugative pili, and these surface structures are really important in basically mediating conjugation. They allow the formation of this kind of like cytoplasmic pore that mediates the transfer of the genetic information from a donor bacterial cell to a recipient bacterial cell. And this conjugation only occurs when the two bacterial cells recognize each other and they come into cellular contact, basically. Now, conjugation is largely driven by the large conjugative plasmids. So plasmids are basically these accessory genomes. They are circular DNA molecules, and they constitute an accessory genome in bacterial cells, meaning that they don't carry essential genes, they just carry additional or accessory genes that give bacterial cells additional functions. So for example, like antibiotic resistance. In conjugation, it's these larger plasmids that are normally of a low copy number in bacterial cells, in contrast to like smaller plasmids that are of a higher copy number, so there's many more of them in bacterial cells, just because these larger ones, they normally carry not only the accessory genes, but also the genes that are required for conjugation itself to take place. And those are called the conjugative or the transfer genes. And examples of the large plasmids that do this are the F and the R plasmids. They are large and low copy number plasmids that can mediate conjugation. But conjugation is not solely limited to those large and low copy number plasmids, so like the F and the R plasmids. We can transfer other genetic information as well. So there are these things called integrated conjugative elements that are found within the main bacterial chromosome itself. They can undergo conjugation and be transferred in this manner, but the smaller and the higher copy number plasmids, they can move by conjugation, as well as certain genomic regions in that main bacterial cell chromosome, and the whole chromosome itself can move by conjugation, basically by exploiting the machinery carried on those larger, lower copy number plasmids or the integrated conjugative elements. So as I said, this is such a powerful mechanism because basically any DNA or genetic information can be mobilized by conjugation and can therefore move between bacterial cells. And some other reasons why it's more powerful, say, than transformation and transduction is because it's what we call replicative. And what we mean by it being replicative is that it's not as if the genetic information goes and leaves the donor bacterial cell and then only stays in the recipient. The donor will firstly replicate the genetic information that's going to move, and so it will keep that genetic information, and the recipient will acquire it as well. So both of them will have the same genetic information. It's not as if the donor will lose it. That recipient cell, the bacterial cell that receives the new genetic information, it will stably inherit that genetic information. When the genetic information is transferred in conjugation as well, as I said, it takes place through this cytoplasmic pore, basically like a bridge between the two cells. So it's not as if it's going to be exposed to the external environment and potential enzymes, so nucleases that might break it down, or heavy metals that might, affect the genetic information being transferred. So it's basically just like a safe way of transferring the genetic information, and conjugation is also quite a fast process as well. So all of those contribute to it being this very efficient and this very powerful means of horizontal gene transfer. And in the context of antibiotic resistance, it therefore doesn't take very long for bacterial cells to acquire resistance to multiple antibiotics. And you can imagine a situation where you have a bacterial cell population and one of those cells is resistant to, 4 antibiotics, let's say. And when you're treating that patient, you might just apply one single antibiotic. So you'll kill all of the bacterial cells except that one remaining bacterium. And that's remaining because it's resistant to the antibiotic that you're using. However, because it is carrying a plasmid with resistance to multiple antibiotics, you're not only applying a selection pressure correspondent to a single antibiotic resistance gene, but the 3 others at the same time. So you're basically indirectly selecting for multiple antibiotic resistance genes, and so as we talked about previously, that bacterium will start to proliferate, divide, expand its population size, and all of those resistance genes will be passed throughout the population vertically, so by vertical gene transfer, and by horizontal gene transfer, as I said, by all of the mechanisms. But conjugation, as we said, is very powerful and the most efficient. So now we're just going to talk about 2 examples of antibiotic resistance. So the first is something that I assume a lot of people have already heard about. It is MRSA, which stands for methicillin resistant Staphylococcus aureus. So Staphylococcus aureus is the particular bacterial cell species, and what we're saying is that it's resistant to methicillin in the title MRSA. However, it is not just resistant to methicillin, it is actually what we term a superbug, and a superbug is something that is resistant to a lot of antibiotics, and in this case MRSA is resistant to many, as I said, and including those that are last resort antibiotics. So those that we save for the hardest types of bacterial cell infections, making it very problematic, and it can be therefore very difficult to treat. The second example I'm going to talk about is resistance to colistin. So this is another antibiotic, and it's actually another last resort antibiotic. Basically back in 2015, so a while back now, we discovered the MCR1 gene, which encodes for resistance to colistin, and this was found in China, it was found in pigs and in pork products, and it was basically discovered on a particular strain of E. coli that we call SHP45. I don't know the latest figures on this, but it basically spread very rapidly. It's virtually now found all across the globe, and it was first found in human patients in 2017. And even worse than this, we have 6 different variants of the MCR1 gene itself, and we have 2 additional colistin resistance genes. So we have MCR2 and MCR3, and MCR3 is carried on a plasmid that codes for resistance to 18 other antibiotics. 18 others on a single plasmid, including resistance to colistin, which is a last resort antibiotic. It's just crazy how fast it spread, and as I said, how powerful these plasmids can be in them carrying resistance to so many different antibiotics. Now this leads me on to my last point, why are we seeing this rapid emergence of antibiotic resistance? And with colistin resistance in particular, let's firstly start by talking about that. This rapid spread has largely been driven by antibiotic overuse in human patients, and also their overuse in the animal agriculture industry. As I said, colistin resistance, that MCR1 gene, was first discovered in pigs and pork products in China. And after MCR1 was discovered, in the year after actually, so 2016, China actually banned the use of colistin in animal feed because they saw it spreading so rapidly. And that just literally speaks for itself. One of the main ways in which we're going to tackle antibiotic resistance is to stop overusing them. Firstly, yes, stop over-prescribing them in human patients. Like, they should only be given and administered when a patient has a bacterial infection, not a viral infection, only bacterial cell infections. And that's basically to avoid applying an unnecessary selection pressure. If you have a patient who is healthy and does not need antibiotics, but you give them an antibiotic, you're going to be killing susceptible bacterial cells and leaving only the resistant ones surviving, which then have less competition, as we talked about, and they can then grow more rapidly and spread their resistance genes. And then in the animal agriculture industry, we also need to stop overusing them. They're basically added to the feed to speed up growth, to prevent them from getting diseases, but they're used in healthy animals. Like, there's no need to prescribe antibiotics in healthy animals. They should only be used when an animal is ill, and the only reason why they're being overused in the animal agriculture industry is because there's a huge demand for animal products. We need to significantly reduce our consumption of animal products to solve this issue, because that's going to reduce the demand and that's going to stop them being used in animal agriculture. So stopping overusing antibiotics is literally like the primary way in which we're going to combat this issue. In hospitals though, patients should be screened and they should be isolated if it's found that they're carrying resistant bacterial cells, and obviously hospitals should just have like really high hygiene standards as well. Now the last thing I just wanted to say is that we could potentially solve this issue by developing more and newer antibiotics, right? But I don't see that as a sustainable solution, just because we will develop antibiotics, but bacterial cells are always one step ahead of us. They will eventually become resistant to those new antibiotics that we develop. Already pharmaceutical industries are losing interest in investing in new antibiotics. So I think like in 1990 there were 18 companies investing in the development of new antibiotics, but in 2011 there were like 4 of them. This industry is rapidly declining just because antibiotic resistance, the scale of this problem is so huge. And as I said, the only way we're going to solve it is by stopping overusing these drugs. We do not want to end up in situations where we cannot treat patients because we don't have drugs, antibiotics, that are effective. So I encourage you to do more research. Obviously look out for antibiotic resistance in the news, and you yourself as a patient, say if you ever need drugs, like always make sure that you have been appropriately diagnosed and you are getting antibiotics only when you really need them. But as usual, I hope this video was informative, useful, interesting to listen to. So definitely like this video if you enjoyed it, subscribe for more biology with ByHollyG, and as always I will speak to you very soon in another video. Bye!